Hemaglutination Procedure. In this video, we will look at hemagglutination, or the agglutination of red blood cells. We'll use anti-sera, which contains antibody, and red cells, which have the antigens on their red cell surface. Let's begin by looking how red cells and antibody combine to form agglutinates. Place 1 drop of anti-sera A on the left hand of the slide and 1 drop of anti-sera B on the right hand side. The anti-sera contain the IgG antibodies to the corresponding antigens on red cells. Next, thoroughly mix your red cell reagents. Then, place 1 drop of red cell reagent A on the left-hand side of the slide and 1 drop of reagent red cell B on the right-hand side of the slide. Gently rock the slide to mix the red cells and the anti-sera. Agglutinates will form if the antigens on the red cell combined with the antibodies in the anti-sera. This is how we type patients' red blood cells for A, B, and O blood groups. Now, let's look at the agglutinates that were formed. On the left-hand side of the slide, A red cells with the A antigen have combined with the antibody A to form agglutinates. Left-hand side, B antigens on the red cells have combined with antibody B and formed agglutinates. This confirms that red cell A has A antigen and red cell B has B antigen. Let's test a patient, red cells in plasma, for antigen A and antigen B. Place your anti-sera in the corresponding spots on your slide. Mix your patient red cell suspension thoroughly and place a corresponding drop to each side of the slide. In this test, we are testing our patient's red cells for the antigens A and B by using anti-sera, which contains antibody A and antibody B. Agglutinates will form if the antigen and corresponding antibody are present. This is called the forward group test in an ABO typing. Let's look and see. No agglutinates were formed with A or B. This tells us that our patient's red cells do not have antigen A or antigen B. Next, we'll test our patient's plasma for the presence of antibody A and antibody B. We'll use our reagent red cells for this test. Place 1 drop of your patient plasma on each side of your slide. Gently mix your reagent red cells to resuspend. Then place 1 drop to the corresponding spot on the slide. Remember, we know what antigens are on the reagent red cells. We are looking for an antibody in the patient plasma. Gently mix your slide, making sure not to run one specimen into the other. According to Landsteiner's rule, if the antigen is present on a red cell, the patient will not produce the antibody. Conversely, if the antigen is not on the red cell, the patient can produce the antibody in the plasma. This is the reverse group test of an ABO typing. Let's see what we have. We have a agglutinates formed with our reagent red cell A and with reagent red cell B. This tells us that the patient's plasma has anti-A and anti-B, which confirms O blood type.